Yes, see here students. So now we have discussed the mechanism of replication. So with two steps in the previous video. Now, so activation of nucleotides and opening of DNA helix, right? So we have discussed. So now the formation of primer, formation of third step is formation of primer. Let us we start with this formation of primer. Just you can once again remind this one. Nucleotides are actually they are deoxyribonucleotides are in inactive form. They are become active, and here in opening of DNA helix by the help of so which enzyme, that is DNA helicase. DNA helicase we discussed, right? So now the formation of primer. So actually DNA, new strand of DNA is begins with the RNA nucleotides, right? So that is ribonucleotides, not deoxyribonucleotides. The small segment is formed, so that is at the beginning. See, for example, when it is open strand, so why like fork, replication fork is formed here. So I will take up here. This is what the, yes, this is what the diagram. So I will take it. Right. So these are the two strands of DNA which are get opened in the second step what we discussed. So these are the two strands here. For example, it is a unbroken strand, and but it is linear now. So it's said to be linear. So now this is completely open strand. This is what we call the this region we call it as a ORI site. This is called the ORI site, right? ORI site, the origin of replication. So where the replication is begins, right? So this what happens? So due to presence of what you call the helicase, so these are also get opened. The hydrogen bonds are broken and it is also opened. So the simultaneously the what happens? Formation of primer is takes place here. So consider here, this is the third end. This is the fifth end here, right? So otherwise you can take this side as per in the book only you can take. So this is the, so I'll take here. See, this is the fifth end. This is the third prime, right? Fifth prime of this strand is here. Third prime is here. So this will be opposite, that is third prime. And this will be fifth prime, right? So these are the parental strands, also called, yes, these strands are referred as templates, or parent strands, right? So this you can mention as A and B. So A and B are the parent strands here. So as we studied in the semi-conservative method of replication of DNA. Now, so here the RNA primer, so that is primer is formed. Primer means here it's a small segment of RNA, small segment of RNA. So because it is made up of ribonucleotides. So ribonucleotides are joined, some about 8 or 10, some minimum about maybe 20, right? So few ribonucleotides are joined. So and it forms the, yes, a structure that is called, yes, this is what we call primer, right? Few few nucleotides, ribonucleotides are joined to form a, so I will take it as very small here. So with some nucleotides, this is what the primer, I will consider it as a primer, right? So this RNA primer, so has the open here, the one end is with the OH hydroxyl group, that is at the third end, and this is at the fifth end here, right? See, the functional group OH, that is hydroxyl group, so which allow to join the next nucleotides, right? So for that sake, so there is a, yes, there is a primer formation which takes place. So primer is begins at the fifth end, right, from fifth to third end, right. So we can say that is third end, at third end, and this is the fifth end here, right, fifth end. See, single strand it is, right now I will consider it as like this. So this is the, yes, this is the fifth end, and this is the third end, third prime. So at third carbon atom there will be a OH is open, which is helps to join the other nucleotides, deoxyribonucleotides. Then deoxyribonucleotides are formed like this, and which makes the continuous strand here. So which makes a continuous strand, formation of a continuous strand towards the, yes, the fifth prime to third prime, right. So this is even there is another step also we can take here. So this is what in this direction, yes, it's a continuously formed strand. So this is the primer, and this is the, yes, new strand which is continuous. So this is what we call the new strand, new strands, and it is continuous. It is said to be continuous, hence it is called, yes, leading strand, right. So another one is, so actually the primer is also begins with the, yes, the fifth to third direction. So here fifth, actually what happens, there is no third open end, so that's why. So what happens here, it is not become continuous, right. So there is a primers are there in this pattern, primers are formed, right. So first means in many steps we can take. So there are many primers are formed. These are called the RNA primers, right. So this RNA primers or helps to continue the next what you call polymerization of deoxyribonucleotides. So this is what here. So I will take it by this color. So you can notice the color. So this is the new strand, and here, so for this one, so I will take here fifth end this side, and here third end for this side. So this one is, I will show here, this is the what we call, so this is fifth end, fifth one, and here third. So in this way the small segments are formed here as a primers. There are many primers are formed, and to continue that the deoxyribonucleotides joins in this way, right. This way deoxyribonucleotides are joined to form a, yes, new strand. Right, in this way there is a segments are formed now. These segments are called Okazaki, Japanese scientist name, it is Okazaki fragments. So it is become discontinuous. So this discontinuous strand, this is not continuous strand, discontinuous strand. So this due to discontinuous strand, so we call it as a lagging strand. Both are new strands only, both are new strands, right, both are new strands, but it is called lagging strand, right. So always the new strands are formed in fifth prime to third prime, so already fifth prime is there here, so it is not started with from the fifth side, right, fifth side. So because complementary is become third to fifth, it will not started, it's a formation of new strand, formation of complete formation. So that is what fourth in the fourth one you can take the step. So that is, yes, see here the formation of primers where takes place. So primer is nothing but a small segment of RNA which has few ribosomal what you call nucleotides, ribonucleotides, right? Ribonucleotides which are formed here and which provides the functional group, that is hydroxyl group at third prime, right? Third prime. So then it is joined with the help of with the other nucleotides, deoxyribonucleotides activated in first step. What we learn, no, activated that as a substrates, what we call substrates, right? So that substrate is joined, that is called nucleotides, deoxyribonucleotides. It may be adenosine nucleotides or guanosine nucleotides, deoxyguanosine nucleotides, or cytidine or thymidine. So like these nucleotides are joined complementary to the parent. So this is what we call template. This is what the template strand is there, no? So which nitrogen base is there, so for that complementary is formed, right? So then that's why we call it as a new strands are said to be complementary. New strands, these are the what we call both this leading strand and lagging strand are considered as here the what you call complementary strands. They are said to be complementary because adenine combines with adenine combines to the thymine, adenine thymine, and guanine cytosine. So in this way there is a complementary strands are formed. So one is become continuous strand. So that is what the fourth step what we have taken is formation of complete strand. During formation of complete strand, so always this parent strand is with third prime to fifth prime. Third prime to fifth prime, in this direction it is there, right? So this is what this direction, and here the new strand is begins with fifth prime to third prime, new strand. So this is what opposite we can say. Always it starts with the new strands or complementary strands are begins from fifth prime to third prime, towards the third prime. In the same way, the lagging strand is also become completely formed by replacing the RNA primers. These RNA primers are replaced by DNA, what you call deoxyribonucleotides. These are replaced by deoxyribonucleotides. So by this these are also removed, these are get removed here, right? So in presence of polymerase, right? So here also polymerase will sliding on this way, it will be sliding, polymerase will be sliding and it starts a polymerization function, right? So and one more here, the what you call a ligase. So we can say that is ligase. So this ligase helps here and that joins the these Okazaki fragments. Ligase helps to join the Okazaki fragments, and polymerase, one more enzyme, polymerase, which makes the gap fulfilled, right? Means this RNA primers are removed and instead of that deoxyribonucleotides are joined. By this this will become also continuous later on. Okay, so at beginning when the complete formation, before the complete formation it was discontinuous, hence it is called lagging strand. At the beginning only it is started with completely forms, so that is why we call it as a continuous strand. Hence we also refer it as a leading strand. This is continuous strand is called leading strand. So both leading and lagging strands are said to be complementary or the new strands, right? So here as in the fifth step, removal of primers by DNA polymerase, which are replaced with the help of deoxyribonucleotides, as I explained. Next one, the proofreading. See, proofreading or repair. So that accuracy is checked with the help of one of the DNA polymerase subunit, that is epsilon subunit and nuclease. These two enzymes are involved in the accuracy or what you call proofreading or the repair, right? So this is what the replication of DNA, the mechanism of replication of DNA, so which makes this fork like structure, that is what Y shaped, inverted Y shape or Y shape we can consider. So this when you observe this one complete formation, again this will be continuous up to the end, it will be continuous, it will be forms. So that means it forms the, so next step is like this, right? So this strand is continuously growing, and again so this strand forms the segments, Okazaki fragments, again that is forms the that is also becomes replaced by DNA polymerase. So and by the ligase it will binds, right? Ligase can join the two fragments, or each and every fragments are joined, right? So which makes it continuous, right? So in this way two new strands are formed, and that is become completely formation of this structure. So here one strand of this one and another strand, so these are become these two strands. In this way the two strands are formed, right? So it's for understanding we have made here steps. So even some are made 7-8 steps, or only 2 or 3 steps we can understand. For understanding purposes we have done the activation of nucleotide and opening of DNA helix, right? Opening of DNA helix and formation of primer. So there is a enzyme called primase. So here we can mention primase. Already the functions of that enzyme we have explained in the previous video. So once again you observe that video. So that is primase helps to form a small segment of RNA at the beginning of this one. So formation of new strands. So it provides the OH group, okay, that is a functional group to join the next substrates, right? So next formation of complete strand. So the formation of complete strands involve the DNA polymerase and even the ligase, right? So ligase, DNA polymerase, so are also involved. Removal of primers. So here also DNA polymerase is involved, and that is proofreading involves these enzymes, that is epsilon subunit, that is polymerase, one DNA polymerase, and one more is nuclease. So these are also enzymes which are involved, right? So this is about the mechanism of replication. So with both video we can observe at a time, then you will understand, right? So go through the textbook line by line. So if you have any doubts you can comment, and so I'll make the another complete video, okay? Thank you students.